Awesome. Hi, everyone. My name is Michelle. I'm a master's student in the Land and Atmospheric Science program. I'm a research assistant for Dr. Griffis, and we are part of the biometeorology group. Today, I will be presenting on our agricultural bacterial inoculant mesocosm experiment. So I'll start off with a little bit of the importance of our research. In North America, agriculture is the leading source of nitrous oxide emissions. And this is from synthetic nitrogen fertilizer that's added to agricultural systems, which leads to leaching and runoff, which causes water pollution and also causes air pollution through emissions. So why are we so worried about nitrous oxide? For one, it's an ozone depleting substance. So in this graph, you can see its ozone depleting potential compared to a lot of other substances. So it's basically inert in the troposphere, so it doesn't react with anything until it reaches the stratosphere and reacts with ozone and can diminish the ozone layer there. Additionally, it also has a really high global warming potential of about 300 times that of carbon dioxide on a 100-year time horizon. So even though the concentration of it in the atmosphere is a lot less, it still has a really high potential to cause radiative forcing. So this brings me to the product that we're testing called Proven by a company called Pivot Bio. It's a microbial amendment that you add to the soil. And the idea behind it is that it colonizes on the crop root and forms a mutualistic relationship with the plant and fixes atmospheric nitrogen into plant available forms and supplies the plant with nitrogen so that less synthetic nitrogen fertilizer has to get added to the system, which decreases pollution, as I just said. So this microbial amendment has edited genes for one, suppressing enzymatic activity that would allow the microbe to assimilate the nitrogen itself. So instead, it excretes it out into the plant, and so the plant can take it up. And for two, it allows the microbe to keep fixing nitrogen even when the soil nitrogen concentrations are high, when other microbes that weren't edited would stop fixing nitrogen. So this is a picture of the greenhouse facility that I work in. It's right here on the St. Paul campus at the plant growth facilities. It has 6 big soil columns. It's the big thing in the silver boxes. It's filled with soil and it supports plant growth. We are growing corn out of it right now. And this is a really reproducible setup because we can control like the light and the irrigation that the plants get, as well as it's a climate controlled greenhouse, so we can control the temperature in there as well. A little bit about the experimental design. So it has gas flux chambers installed on every soil column. So the one on the left is closed. It's a little darker than I would like, but the one on the left is closed and the one on the right is open. So it takes hourly fluxes from the soil and closes for 10 minutes and then goes to the next chamber. So it takes hourly flux measurements. We also do plant samples at the end of the season for carbon and nitrogen, and we do weekly water and soil samples and send those in for chemical analysis as well. So these big chambers are gas flux chambers that take air from inside and they go to a gas analyzer. And this one does carbon dioxide, nitrous oxide, methane, and water vapor measurements, as well as we have ammonia measurements on 2 of the soil columns. So we have gas flux chambers for that as well. So the 2 things that we are really looking at is yield increased with Proven, as well as how nitrous oxide emissions are diminished through the reduction in fertilizer rate. So these are some preliminary results. We've done a few different setups, but I'll just go through one with you guys just for the sake of time. We did a 40, 60, and 80% rate of fertilizer via urea. And so a 20, 40, and 60% reduction of a non-limiting 100% rate that a farmer would typically put on their soil, which we said was about 202 kilograms of nitrogen per hectare. And so this is a graph of the average yield for each fertilizer rate for 2 seasons. And so 2 soil columns were dedicated to each fertilizer rate. One of those was treated with proven, and then the other one was not. So we had a control and an experimental variable for each one. So the ones in the green boxes are 40 and 80% nitrogen. The proven is the green one, and that had a higher yield in 2 of the rates. And then in the 60% rate, the proven did not have a higher yield. So I've ran some statistical significance tests on these, and I have found no significant yield differences between proven and non-proven treated soils. But this is definitely still an ongoing experiment. I'm still a first-year master's student, so this is something that I will be continuing to look at. And I'm happy to do my best to answer any questions. No, so those are standard air bars. The yield is a little bit tricky to do this with because there's only like 12 data points. I only take 1 data point for yield per rhizotron, per soil column. So that was only over 2 seasons. So it's 12 data points. So I decided it would make more sense to do like a standard error bar rather than a standard deviation error bar. So that those error bars don't necessarily represent like a statistical significance. But I'm still looking into it and trying to find the best way to look at that.
I got a same question.
That's a good question. Proven has actually offered to do that for us and to test like our plants to see the nitrogen uptake and to see if it's really going through the roots and the plant is getting more nitrogen. We do carbon and nitrogen plant analysis at the end of the season, though I haven't exactly looked at those results yet, but we are getting those tests. Somebody said something on Zoom.
Yeah, I have a similar question like the first question. You have 6 boxes, right?
Yes.
You have 6 containers.
Yeah.
And you have 6 bars in your graph.
Yeah.
So 1 bar was for each container?
Correct. Yeah. So we had 3 fertilizer rates. So 2 soil columns per rate because we have 6 soil columns.
Okay.
And then one of those was treated with proven, and then the other one was not.
And how did you get the error bar? I missed on your explanation.
Yeah, so it was a standard error bar rather than a standard deviation error bar. So it doesn't necessarily represent like the statistical significance, just because the amount of data points from the yield is so low that it's difficult to run significance tests, but I'm still looking into it.
But was the error bar of the measurements in one container, or was the error bar across the container? You cannot have across the containers. You know, the error bar that you got, I'm just trying to understand it. That means you took 5 measurements in one container, and that's the error bar of those 5 measurements?
It's just a standard error bar.
Okay. All right, I'll let go. Thank you.
How do you have an error bar for one data point?
Yeah, I don't know. I guess I don't really understand. I just plotted it up using the average of the yields from each thing. So I have 2 data points, and then there's error bars for those 2 data points. I guess I'm not sure exactly how it did it. I just put it on there.
Okay, thank you.